All right, so good evening and welcome to this evening's webinar. In front of you is the roadmap for this evening. Ms. Ninja and I will take you along with, and yes, you probably see along the lower edges of the camera little outline of fur. That is Ms. Ninja. She is on her little binder that I've put on top of the keyboard so that she does not interfere with the slides and everything like that, but can still be close and join us. In addition to the roadmap there in front of you, I need to take care of a few housekeeping items. As always, the chat box is a way for you to communicate with me, and I will try to catch questions as we go along through the program. Feel free to drop those questions in the chat box, and I will answer them as I can. As always, I do stay a few minutes afterwards in order to catch any questions or after program comments that there might be. The chat box is also a way for me to basically take attendance and provide you with emails that will first contain a post-event quality survey. That'll go out sometime within the next 24 hours. And if you fill out the survey, and you do need to fill out the survey, then you are eligible for a second email, which contains a link for your certificate of completion. This evening's program is a professional development opportunity for educators here in the United States, grades K through 12. So if you do fill out that post-event quality survey, and it only takes you about 5 minutes, 10 questions, then you're able to get that certificate of completion, which you can then show to the folks who are in charge of professional development credits and relicensure in your school district or at your school. Because this is a professional development opportunity for teachers, I will talk a little bit about instructional activities and how they relate to the topic that I have selected for this evening. So if you're here hoping for a super technical talk filled with lots of research jargon, I'm afraid I'm going to have to disappoint you. This is intended to give an overview of the research that's being done on the International Space Station, these particular two topics that I have, or two projects that I've selected, and how that ties into the material and the curricula that the United States teachers must cover in their classrooms. So just want to be upfront and let you know that this is more geared towards education, teaching K-12 students, as opposed to any super technical, in-depth research talk. Let's see. If you are interested in getting a copy of my slides, I always make them available through what I call a resource email. Now, I know some of my colleagues provide the slides for download immediately after the program. I do something a little bit different. I'm also recording the program, and it takes a little bit of time for the recording to buffer. So between 1 and 2 weeks from tonight, if you ask for the resource email, you will get a third separate email. It's totally separate from the post-event quality survey or the certificate of completion. There'll be a third email that you will get that will contain that recording, will contain a way for you to download my slides from Google Drive, a PDF version. I include links for all of the video links, both before and during the program, and then a few other goodies that I throw in. So I see some chatter in the chat box, and I'm going to just quick scroll through. I think that's most of you just asking for the resources, which you can just drop an email in the chat box with a little notation that you would like the resources. If you're not comfortable putting your email in the chat box, you can hover over my name and create a personal private tab and send me that information, so that way then you let me know that you'd like the resources, but that information is not in the general chat. I'm also seeing some folks indicating that there's a little bit of an issue with the audio. Most people are saying that they can hear me okay. If you are having any audio or video difficulties, it could be on my end on the connection, but it's possible that something's happening en route. Usually the best way to change it or to fix it is what you're seeing in the chat box. So folks are saying log back out and log back in. I am recording the program. Usually any issues with connection do not catch up in the recording. So if you are having trouble with the connection, by all means ask for the resources, and that way you can get at least a copy of the recording, and that way that should hopefully fill in whatever you can and cannot hear from me in the live broadcast. Okay. right. For those of you who don't know me, my name is Dr. Ann Weiss. I am an education specialist at NASA Langley Research Center here in Hampton, Virginia, which is southeast corner of Virginia, practically on the Atlantic Ocean. We are about to get our third snowstorm this coming weekend, which is interesting because Virginia, I had a friend a long time ago told me, was a native here, Virginia, said, down here at the beach, we almost never get winter weather. And now in the span of 2 weeks, we're looking at our third snowstorm. So I'm going to be giving that friend a little bit of a hard time about the fact that we don't get snow here in the southeast corner of Virginia. But anyway, I am a research scientist by training. So although I am going to be focusing this particular program from the educator standpoint, teaching K-12 students, I am going to be able to include some itty-bitty overview of the research science because I am trained in those methods, both cell biology and molecular biology. And that was my original career until I switched over to education about 2 decades ago. And I've taught everything from middle school science up to undergraduate anatomy and physiology. So I am super excited to be sharing this particular research projects with you and how they factor into our plans for going to the moon and eventually on to Mars, which was the subject of one of the videos I showed prior to the start of this program. So let's go ahead and dive into the main material of the course. What I want to do real quick is just kind of set the stage for the environment. We're going to be going to the International Space Station, obviously not actually, but virtually, kind of in virtual field trip. And we just recently celebrated a milestone. Of course, the Russians and the Soviets have, for a very long period of time, focused on long-duration spaceflight on their space stations like Mir, which the United States and Russians partnered on in the 90s. But starting in 2000, and specifically 2001, we've had humans on board the International Space Station, which is a collaboration of a variety of nations that have developed this platform in orbit, low Earth orbit, and we've been fortunate to have a human crew on board ever since, I believe it was November of 2001. So we are now celebrating 21 years of continuous, unbroken human presence in space, which means pretty much that the class that is graduating from college this year, they were born just about the time that the first crew, Expedition 1, arrived at the International Space Station. So we have a generation of students who have known, even if they were not aware of it, they have lived in a society where there's been a human living and working continuously throughout their entire lifespan. So that was something that we celebrated last year and continue to celebrate quite a bit. And there's a number of reasons why NASA invested in the International Space Station to create it. One of it was to develop a space economy and to include people or companies or partners in the process that may not have necessarily been traditionally included. So for a very long period of time, it was governments that were launching astronauts into space. And with the International Space Station, that has provided an opportunity to bring commercial partners into the program. So when the decision was made to retire the shuttles, there was a need for launch vehicles from the United States. And so NASA engaged in a call for proposals that included a variety of companies, but was eventually down selected to Boeing and SpaceX to develop spacecraft that would carry humans from Earth to the International Space Station. We have a number of other agreements for resupply of the International Space Station, which includes SpaceX, of course, but also Northrop Grumman Orbital ATK. So the International Space Station, one of the ideas for its creation was to stimulate this space-based economy, initially by having commercial partners service the International Space Station, but it's very quickly going to extend beyond that to companies that will be building their own space stations, their own private space stations in orbit. Perhaps we might have space hotels. We might have a growing city in orbit around us at some point. And the International Space Station is basically that idea to kind of stimulate and encourage that development of low Earth orbit beyond just government entities. And so if you're interested in knowing a little bit more about the NASA Commercial Crew Program, I encourage you first of all to attend some webinars from some of my EPDC colleagues, to include Dr. Samuel Garcia and also Stephen Smith. They at least once a month will do, either one of them or both of them, they will do what's the latest and greatest with Commercial Crew. So I totally encourage you to check out their webinars if you'd like to know what the latest and greatest is going on. Right now we have Crew 3 up there. They will be up there until about April or May. So there's plenty of information at the link there that's on your screen that'll take you, first of all, to the social media, the Twitter, the Instagram, the Facebook, stuff like that, so you can follow along what's actually going on with the Commercial Crew Program and Crew 3 as they're at the International Space Station. But the toolkit, the Launch America Toolkit, also includes a variety of educational activities split up between grades K-5. There's a group for instructional activities for that grade level, but also then grades 6 through 12 for the advanced students. And so you can scroll through, and it includes things like coding sim, which allows students with mathematics, graphing, algebra, to basically plot how they would navigate either the Boeing or SpaceX capsule to docking with the International Space Station. So it not only includes the math, but there's also a computer programming application to it as well. So certainly there's a bunch of science-based activities, demonstrations which show basic physical science properties. Mass versus weight, for example, shows different demonstrations on the International Space Station, and then ways that you can incorporate adaptations of those activities in your classroom. So if you're interested in learning more about the Commercial Crew Program, I encourage you either check out those webinars that I mentioned or this Launch America Toolkit that we update depending on which crew and mission is up on the International Space Station. Let's see, can you send a program link to email? Sure. Okay, so I will, actually I'll try to drop the link to our full schedule. And that reminds me, actually, I forgot to mention this during the housekeeping items. I'll try to remember to put a link for all of our webinar schedule in the chat box at the end of the program, unless somebody beats me to it. But just a programming note there, we will only have one webinar next week. That'll be Dr. Barbie Buckner on Monday evening, because, and some of you are probably well aware of SEEK, the Space Exploration Educators Conference, will run February 3rd through the 5th. We are all the specialists, we are all presenting either in person or virtually at that conference. And we're actually having a team meeting a couple days before the meeting. So next week there will be very, very few EPDC webinars, but we will be back February 7th. So somebody's already gotten, Sylvie, thank you very much for putting that in the chat box. So for those of you who are interested in the upcoming webinars, including looking for those CCP Commercial Crew Program webinars by Dr. Garcia or Stephen Smith, you can check out that link that Sylvie just dropped in the chat box. Thank you very much. So the Commercial Crew capsules, which at this point is primarily SpaceX, we're still awaiting Boeing to solve the challenge or issue that they had, and we're still waiting for them to do their demonstration flight before we certify them to carry astronauts. So primarily when we talk about rodents and astromice and how they get to the International Space Station, they're generally hitching a ride on the SpaceX capsule, whether it be the Crew Dragon along with the astronauts or the Cargo Dragon, which of course is an unmanned cargo ship that gets launched several times a year to the International Space Station. And Patrick has been kind enough to drop the link into the chat box for the SEEK conference if you're interested in learning more about it. I have been there twice in person and attended virtually last year. It is great for those of us who love the space program and incorporate those things into our classroom. And oh yeah, usually there's tours and astronauts who speak as well, because it's in Houston. It's Space Center Houston, which is the visitor center. So I highly, highly, highly recommend that you check that out if you don't already know what the buzz is about SEEK. On the screen there, you see the hardware system, the three pieces of the system that we use to transport mice to the launch vehicle, then from the launch vehicle into the International Space Station once the cargo ship docks, or the Crew Dragon docks with the International Space Station. And then once they've had a chance to hang out in the habitat on the International Space Station for a couple of months, then they get packed up and they come back down in the transporter. In order to facilitate transfer between the habitat, which is on the International Space Station, and the transporter, which is their custom accommodations for getting up and down from space, we use the animal access unit that helps the astronauts transfer the Astro mice from one to the other with hopefully no mice escaping and swimming through the International Space Station with an astronaut chasing after it, trying to catch it before it gets too far from the habitat. At any rate, and yes, Seek is also a virtual / in-person, so it's a hybrid event again this year because of the Omicron surge. Talk mice and a cat will show up. Oh yes, even though she generally tends to keep her attention to the toy mice. But yes. All right, so for those of you who are interested, particularly if you teach the upper grades or are university professors teaching introductory biology or anatomy, physiology, and might be interested in a little bit more in depth as to the actual nitty-gritty of how we get the astromice up and down, here's your con ops for the rodent research on the International Space Station. What you'll see, once I get my arrow here, once I figure out where my arrow is, there it is. So you start over here on the left side. So they're packed up into their transporter, usually about 10 to 20 mice per transporter, along with all of the materials, food, water, etc., that they might need for up to 10 days. Astromice and any animals that are going up for research on the International Space Station are usually the last loaded onto the space capsule to try and minimize the amount of time that they are in the transporter with that limited supply of food and water. But assuming that all goes well, launch goes as according to plan, then they are going to be on the ship approximately anywhere between 1 to 3 days before the capsule docks at the International Space Station, at which point up here along the top, then they're going to get transported into the habitat, which is where they'll stay. And they adapt very quickly to the habitat and adapt to weightlessness fairly easily. So while the launch might be a traumatic experience, of course it's traumatic for the humans as well, the mice tend to adapt fairly rapidly to living and floating in space. In fact, I've got a link somewhere where they've got mice and they're running laps, or however you run laps in microgravity. They've got these special metal bars that they can grab onto, and the mice is just using those metal bars to zip themselves around the cage. But at any rate, obviously while they're up there, there's work to be done. So the middle part here kind of shows how some samples might be obtained in orbit, and this is a full-scale lab with a glove box, clean box, etc., and able to take blood, urine, and tissue samples from the astromice, and then pack them up. Sometimes freeze them, so there's a freezer there on the module and everything like that. Of course, at some point, astromice need to come home just like the human astronauts, so they are then transported, or excuse me, they're then put back from the habitat back into the transporters, packed back into the capsule, and then a splashdown. And certainly by the time they splash down, the scientists want to get those tissue samples as quickly as possible, and the live mice as quickly as possible. So usually there's a helicopter waiting, and will bring the astromice post-haste back to land and get them to the investigators so that they can do their data analysis. All right, so Although there have been space stations in the past and stuff like that, the International Space Station right now is the premier orbiting platform right now. It will be joined shortly, as I said, by some private space stations, but for now, the International Space Station is it. It is a unique situation, only place where we can test things in microgravity, and that allows for at least the United States portion of the lab. Remember, it's an international collaboration that built the station. The United States portion of it is designated as a national lab. Here in the United States, we have a system of national labs. If you caught my slide introducing myself, you will notice that I worked at Los Alamos National Laboratory. That's one of the 8 that's run by our federal government, and the portion of the International Space Station that's run by the United States is technically considered the 9th national lab. So that is pretty much where we do a whole range of technology and science experiments. And one of the other things that the International Space Station is helping, I mentioned a bit about the space-based economy and opening that up for commercial partners, private industry, but another important objective for the space station is to get us ready for the Artemis program, which was the subject of one of the videos I showed at the start of the webinar, before the start of the webinar this evening. Artemis, in short, is our next human space program. It's going to first take astronauts to the moon. We're going to develop a moon base, so it's going to be more in depth and more involved than the Apollo flights were. So astronauts will now be spending weeks or months on the surface of the moon once that moon base gets up and running. And we plan on putting that on the South Pole, where there's, thanks to some satellite imagery, a fair amount of frozen ice beneath the surface of the moon. In addition to the moon base, there will be a space station in orbit around the moon. That's going to be referred to as Gateway, and that will provide the basis of operations for getting us up and back to the surface of the moon. And at some point, as we've learned to live and work on another planetary body, our moon, then we will stage and then set our course for Mars in a couple of decades, perhaps 2030s, perhaps 2040s. So living and working on the International Space Station, learning how to do that has been foundational for us to even attempt living and working on the body or on the surface of another planetary body. In this case, it'll first be the moon, hopefully eventually Mars. So International Space Station is incredibly important, and you can kind of see those 5 areas of research or inquiry that most of the experiments or technology demonstrations have fallen into. And this also kind of gives you a little bit more in depth how those 5 areas kind of fall out in terms of the different scientific and engineering disciplines. Just about any area of scientific inquiry or engineering innovation has been at least touched upon at one point or another during the 21 years that the International Space Station has been in operation. Now, of course, the first part of building the International Space Station was just simply getting it up and running. But fairly soon after the main structure, the basic structure of the International Space Station was in place, science has been ramping up. And one of the reasons the Commercial Crew Program is so important is it allows us to ferry astronaut crews up and back to the International Space Station much more frequently than we were able to when we were depending on the Russians to get up there. But it also allows us to get more people on the International Space Station. So each expedition crew is approximately 3 individuals. With SpaceX, that's usually 4. But with the Commercial Crew Program and our ability to get astronauts up to the International Station more frequently, we're able to maximize not only the number of astronauts on board to approximately 8 or 9, but also to ramp up the amount of science. So it's kind of been a steep curve, starting out slow with research, but then expanding very quickly. And as of towards the end of, I believe it was 2020 going into 2021. So during that 20th year celebration, the statistic was something about 3,200 experiments had been conducted on the International Space Station up to that point. So obviously it's been a year, almost a year and a half since then. So you can just figure that number continues to grow. And one of the things that I truly enjoy pointing out as part of focusing on International Space Station research is, okay, yes, we're building a space economy. Yes, we're getting ready to send astronauts to the moon and on to Mars. But for the vast majority of us, we're not going to the International Space Station. We're not going to the moon. We might want to. I certainly wouldn't mind getting a chance to ride into space. But the vast majority of us here on Earth are not necessarily going to have that opportunity just yet. Perhaps later, as the commercial partners get ramped up, private industry gets ramped up, maybe. But right now, the likelihood that I'm going to get to go into space is rather low right at this moment. But the idea of the International Space Station is also to provide benefits back to humanity. So while we're doing all of this research with the idea of preparing the astronauts to send them out further and further into the cosmos, one of the most important things here as NASA is how can we use what we learn in the space program and spin that off and deal with challenges and situations down here on Earth. And certainly with biomedical research that we're doing, that's the focus of tonight's webinar, there's fortunately it's very easy for me to make the connections between what's going on the International Space Station and how that might benefit you and me at some point in the future from a medicine and biological research perspective. Okay, so I think everybody's having okay with the video. So we're focused obviously going to be human research, and once again I'm going to take a slight sidestep and talk a little bit about instructional resources and make you aware of a second toolkit. So I made you aware of the Launch America toolkit for the commercial crew program. This one, STEM on Station, this is a toolkit that brings all of the excitement and wonder of the International Space Station research and engineering projects, bring them down to your classroom. So I've decided to highlight specifically the collection of resources that specifically address human research, because of course that's the topic, overarching umbrella topic for this evening's webinar, and that's what the link there is, will take you directly. However, there are tabs across the screen. I've kind of provided a screenshot, and I think it's this one here is Celebrating Station Science Home. That will take you to the home page, which then you can access the tabs for all of the 12 months that we focused on International Space Station research last year. So if you're interested in computer science, we have a tab for that. If you're interested in mathematics, we have a tab for that. If you're interested in astrophysics, physical science, material science, we have a tab for that. So in this case, I'm simply focusing on the one that's relevant to tonight's topic, but do want to make you aware that certainly if you teach a slightly different subject area other than biology or life sciences, there are resources available for you to tap into that particular discipline's research on the International Space Station. And just like with the Launch America toolkit, these are divided by grade level, so there certainly are elementary activities and middle school grade activities and high school activity grades. Also, there's some social media there that you can keep track of what's the latest and greatest research that's going on the International Space Station. So those two toolkits provide a massive amount of NASA resources, instructional activities, hands-on demonstrations, multimedia, so videos. So if you like the videos that I was showing before this program started, there's more of those there at those two toolkits. So I highly encourage you to check those out. Because we get such a diverse audience, I don't necessarily go into any more detail with the toolkits at this point, because I know everybody's a little bit different and has different needs in their classroom. So coming back to research, coming back to life science, coming back to the topic this evening, our astro mice, talk a little bit about something that doesn't always get a lot of attention when we talk about science, and that's the ethical considerations. How do we make sure that the animal research that we're doing is humane and is ethical, which hopefully minimizes as much stress, anxiety, or pain to the animal as is possible. Okay, so I'm going to step back and do a sort of like little basic research design here, okay, which is something I had to do when I was at Los Alamos, but certainly I had to do much more of it when I was actually at Arizona State University working on brain chemistry of the brain. We call it neuroendocrinology, because my project actually used Siberian dwarf hamsters, so not mice, but I had bonded pairs of Siberian dwarf hamsters that I was working with in trying to understand what's the connection between the brain chemistry, stress, and depressive symptoms that would show up in the Siberian dwarf hamsters. And so when we think about research design, particularly with animals, but this is also true when we talk about research with humans, we want to try and minimize the discomfort or pain to the subject as much as possible. And so a lot of times we'll opt to do research projects in the test tube, what we call in vitro, or in nowadays we're able to do like tissue chips, tissue samples on a chip, or cell culture. And so that's usually preferable, at least to start, in terms of understanding how a process in the cell might be working. It's not necessary to have the whole animal, so let's go with cell culture. So that's usually where a lot of basic science research begins, and a lot of research design ends up selecting cells or tissues. And in fact, on the International Space Station, they have a whole cell culture setup. So they've got the clean hood, they got the materials to do sterile work. They have the incubators and all of the setup that you might find in a traditional cell biology lab, like the one I worked at Los Alamos or in a university. They have all of that on the International Space Station, obviously adapted for the weightlessness environments, but it's an incredibly sophisticated setup. So that would be the first choice. Can we do something with the tissue chips? In fact, there's a project on the International Space Station right now, kidney cells grown on a chip. And that's what they're doing. They're not using animals at all for that particular project that's looking into how kidney stones crystallize. But at a certain point, cells and tissues can only take us so far. A whole organism is not necessarily the sum of a couple of billion cells. There's a high degree of interdependence and a number of things that come into being once these different tissues, these different organs are now working together to keep the subject alive. And so while a lot of the science research we might be able to do in the test tube in vitro or in cell or tissue samples, at a certain point we need to bring organisms in, and this is true if we're talking about pharmaceuticals. If we want to know how is a drug going to work, well, yeah, we like to make sure that it targets a particular cell process in the cells. That might be where we start with cells or tissues, but there's also going to be toxicity issues. There's also going to be, how do we administer it? Are we going to have, like for humans, are we going to have it administered orally? Are we going to intravenously eject it? There's a number of things that happen that have to be taken into consideration before a pharmaceutical is even considered being approved for human use. And so that's where the animals start to come in. And once again, we try to go with not necessarily mammals right away, but we'll try to use invertebrates. So if you are familiar at all with any of the life science missions that were on the space shuttle, a lot of times they were taking fish, they were taking squids. I think there were honeybees that were taken up. So there's a variety of invertebrates that may be selected for use in an animal research project here in an Earth-based lab, but certainly on the International Space Station. So we try to hold off using mammals as long as possible if it's possible to do everything either in a cell or tissue or an invertebrate. But obviously, at a certain point, particularly if the pharmaceutical is being looked at as possibly being used in humans, at some point mammals, and that's usually rodents, are brought into the equation. Sometimes it might be primates like monkeys or apes or something, orangutans, stuff like that. But for the International Space Station, in terms of space considerations, mice and rats, but usually mice are the preferred mammal to bring on board to the International Space Station. Because of the humane and ethical considerations, everything that's done by NASA, just like in an academic setting, must be run through an institutional animal care and use community. IACUC is what we used to call it. And certainly as part of the review process, and I of course had to put a protocol through for approval for IACUC when I was working at Arizona State University with those Siberian dwarf hamsters, they are going to go over that with a fine-tooth comb. And certainly because we're talking about putting rodents rodents on a rocket, which can be stressful for humans and certainly will be stressful for the rodents. Got to make sure that special accommodations are made so that the rodents are as well cared for as possible, because unlike humans, they cannot give consent. We can't give them an informed consent and tell them everything that's going to happen to them, and they don't have that opportunity to ask questions. What's going to happen? What happens if this happens? So the fact that they don't have the ability to give or withhold consent means that we have to be extra careful when we design our experiments, particularly on the International Space Station, to make sure that they are well cared for. So, as I said, it's not a topic that's necessarily dealt with very often, but particularly for me, it's something that I like to make sure is addressed in the course of talking about this. So I mentioned a bit using mammals and using rodents, and so what I want to do is I want to share this video with you that talks a little bit as to why mice have become such a widely used animal model, not only in Earth-based research, but also in space research. So stand by.
Spaceflight causes many changes in human health. Understanding these changes is important when planning for human exploration beyond low Earth orbit and for managing health and disease on Earth. There are components of rodent biology that are directly related to human biology. Almost every gene found in humans so far has been found in a closely related form in the rodent. We can use the rodent research model for understanding human changes in space. Because changes occur more quickly in rodents, we can study these changes more effectively. On the space station, the rodents will live in a specifically designed habitat. Researchers use both similarities and differences between humans and rodents to gain insight into changes brought about by spaceflight. These insights can improve the health of astronauts and those of us on Earth. Rodent spaceflight studies have already contributed to pharmaceutical development for treating osteoporosis. The knowledge and applications that we gain from rodent research in space are limitless and promising for those in space and on Earth.
Right. So that gives you a little bit of explanation as to why rodents, and specifically why rodents in space. This graphic that's up on your screen right now kind of shares a little bit of why, from a biological standpoint, why rodents, why aren't we not sending primates and stuff like that. Well, as it turns out, if you take a look at the sequence homology, the sequence of DNA in a variety of different species, yes, primates are very close to us, within 98% similarity or homology to our DNA sequence, but mice are 92. And I know when I talk to students and stuff like that, they're kind of astounded, particularly those who are in high school, because they're like, but mice don't look anything like us. Yeah, there's a few significant changes there, but overall, there, as the video said, there is for every human gene at least a copy of some sort of homologous copy of the gene in mice, and so that allows us to be able to justify using animals if the sequence homology and the genes, that underlying DNA architecture is so similar. Then that's what allows us to be able to say, okay, if we create a mouse model of a particular disorder that we see in humans, we can be reasonably certain, because of the sequence homology, that that disease will progress fairly similar to what when it occurs in humans. The maybe only difference may be that it's going to unfold faster in mice, because of course their metabolism is sped up, their life cycle is a little bit shorter than ours. But that is really why mice in particular, rats, and in my case Siberian dwarf hamsters, were and are used in these studies, because of that very close genetic similarity between our DNA and the rodent's DNA. Yep, fruit flies. Yeah, this is always fun for the students, because they're like, "Oh my gosh, there's some overlap between me and a fruit fly," and stuff like that. So this is a fun one. For those of you who are in the upper grades, high school and perhaps undergraduate, I've included the karyotype on the right, that'll kind of show you how the chromosomes look. So the set on the right, remember 23 chromosomes, total of 46, is the human, so that's the color code. And then this very colorful mosaic on the right side shows you where those pieces of human DNA sequence appear to pop up in the mouse karyotype. And so let me scroll back here. There was one question that came up during the let's see, I'm not sure if I get, it was a question that came up during the video about, from Sylvie, do they do some sort of launch testing training for the animals to get them a bit accustomed to the stress? And I answered in the chat box, but I want to kind of go into a little bit more detail. It kind of, the answer is kind of yes or no. Any particular mouse is not necessarily going to go through Astro mouse training before it's launched. But what they've done in the past is they've done a series of tests to see how mice will respond, or whatever animal it is will respond to the stressors. So have there been animals taken up on the KC-135 that those parabolic flight patterns like they do for weightless training for the humans? I believe that they have. I know that there's a very old videos from the 1960s. They took cats, once again, Miss Ninja will approve of that. They took cats up onto the KC-135 and gave the cats an opportunity to be weightless for a period of time. So I believe, although I'm not certain off the top of my head, they did test studies to see how these animals would respond to these types of stressors. But obviously, because animals have shorter lifespans, those particular animals that may have been used in the past for those sort of test studies are not necessarily the same animals that are going up onto the International Space Station on each of these. So we have a base of knowledge that gives us some idea of how to expect how any individual mouse might respond. But as far as I know, I don't think they do any Astro mouse training on each mouse that's actually going to go up into space. Okay. Okay, so let me just make sure. Okay, so Sylvie's asking, so are the colors on the chromosomes representing specific gene sequences? Not necessarily. What they did, and this is from the Oak Ridge National Laboratory, so once again another one of those U.S. national laboratories, what they've done is they simply just decided to give a color to a particular human chromosome. So dark green for, say, chromosome number 1, lighter green for chromosome number 2. And so it's not so much the specific gene sequence, it's just showing you that if you compare the karyotype, the structure of the different chromosomes that are in a human, the homologous or similar sequence corresponding in the mouse happens to be on that difference. So here, which is the mouse chromosome 3, where I've got my thing arrow hovering over the yellow, that's similar sequence to what we see on, I believe it's chromosome 3 in the humans. So it's just sort of allowing to show you from sort of visual perspective that the architecture of the mice karyotype, all of their chromosomes, is not quite identical to the setup or the karyotype of a human, but that there's that sequence homology, that the similarity between the gene sequences is enough that it corresponds to the gene sequence back on the original human chromosome that we gave a full color to. So hopefully that's more clear at this point. All right, so it's 7:45. So the mice are a scrambled version of the human. To some extent, I think that's the best way to think of it. Although obviously, due to the evolution of the two lines, the common ancestor, and then breaking off into the mouse line and then the human line, that there's obviously been changes. Mutations, of course, can happen as a single point mutation, a change in a nucleotide situation, but a mutation can also be cutting and pasting different portions of chromosomes and adding them back together. So yeah, certainly over the period of time, there seems to have been a little bit of mixing up, but I hesitate to say that you should think of the human as kind of like that. This is where we started from, because remember, humans and mice have a common ancestor. So the karyotype of that common ancestor gave rise to those two different karyotypes, and is probably very different than what we see with the humans or what we see with the mice today. So Ileana is asking, how young are the mice when they are taken into space? It really depends on the experiment. So mice typically live about 2 years, maybe 3 or 4 if we're dealing with lab situation type of thing, depending on what the mouse line is, and there are several different mice lines. If I remember correctly, and remember, if they're only alive, if their life cycle is only about 2, 3, maybe 4 years, they're going to mature much more rapidly. So what is an adolescent mouse is going to be very different than an adolescent human. So I believe usually the mice are somewhere between 3 and 6 months old when they go up to the International Space Station. So they're not quite babies, but depending on the research, we don't want them too old, because they'll probably be up on the International Space Station for about 6 months. And for a mouse, a year is about mid-age. So think of like 50s for a human. That's about, that we're starting to get into senior citizen territory with a 1-year-old mouse. So to try and make sure that we're dealing with young mice, and depending on what the research is about, usually 3 to 6 months. If the experiment calls for something specific, the mouse might be a slightly different age. Okay. Okay. Sally Jean asked, do the mice reproduce when on the International Space Station? That's an excellent question, and usually the answer is no. Unless the experiment is really interested in understanding reproduction, no. We keep either all the female mice in one habitat and all the male mice, and there's no commingling. So as we used to talk about it with the Siberian dwarf hamsters, there were pink cages and there were blue cages, but there were no purple cages. So same thing on the International Space Station. Pink habitats, blue habitats, no purple habitats. So yeah, no, they're typically not, they're not allowed to come in contact with each other where they might potentially breed. And the interesting thing, just like we did with the Siberian dwarf hamsters, and the reason we didn't want them breeding, you wouldn't want them breeding on the International Space Station, is that adds another variable into the scientific research. And so I remember from, if you start out a science course, we talk about controlled variables, independent variables, dependent variables. Pregnancy for a female mouse is going to incorporate variables that may throw off the data that we're looking for. So yeah, most times no, we don't as I said, pink cages, blue cages, no purple cages, no mice reproducing on the International Space Station. How animals survive safely to the trip down without spacesuits and behave in microgravity once they're on board. Okay, so I'm going to come back to that because I'm closing in on about 10 minutes and I still haven't had a chance to actually talk about the 2 research projects. So let me come back to that question after I finish the webinar properly. I've left this particular slide up while I was answering questions so you should kind of get an idea of some resources that you can use. This is for my elementary teachers. There's story time from space. I've got the website there for you. Of course, based on the particular topic or book you get an astronaut to actually read. Sometimes it's live. Most times it's a library of recordings. So you can go to storytimefromspace.com to see what upcoming books will be read and whether or not there'll be any live sessions so that you could potentially log in and your class could get story time from an astronaut in space. But as I said, most times it is going to be a recording of an astronaut who has read a book previously. And given the topics that I'm covering tonight, Miss Ninja wanted me to select the mission to Kataria. That's read by Ricky Arnold, who was one of the educator astronauts who was up about 3 years ago now. And then Mousetronaut, which is actually written by former astronaut Mark Kelly, now Senator from Arizona, Mark Kelly, and it was read by his twin brother, I believe, when he was up for the year in space. And so those books are wonderful in and of themselves. Having the ability to have an astronaut read them from the International Space Station just ups the cool level considerably. And Ileana says, loves, thank you, my daughter loves Mousetronaut. Yes, most of these stories are beloved by youngsters, and that's why I said you read the book, it's cool. I have a nephew who loves, he's much, much older, he's about to graduate high school, but when he was much younger, he loved Max Goes to Mars and stuff like that. So yeah, these are great, particularly for the younger students, and of course I always try to make sure that there is something in these webinars for everyone who teaches the various grade levels. First Woman is also fabulous. Yes, yes. Okay, so quickly, very quickly, I'm not really going to go into these slides too much because they're a review, but I put them here so that folks who may not necessarily be life science teachers are able to remember what exactly these particular systems do for us. So the particular research projects that I selected for this evening deal with the skeleton, bone loss, and muscle loss. And so this is just a quick review of what our skeletons do. The primary thing that I want to focus here on as we go forward to talk about these research projects is the mineral storage that occurs. A lot of times our students think, oh, the skeleton is just to give us form and to allow our muscles to move, but it's arguably more important function is to actually store the calcium or the magnesium or the phosphorus and have it be available for when other parts of the body might need it. So when we talk about, drink your milk, your bones need calcium, it's actually for the bones, yes, for part of bone production, but calcium is also used for regulation of muscle contractions. You can't have a muscle contraction without calcium. If I remember correctly, I believe calcium is part of the blood clotting process, so you need calcium for that. Being that I'm a neuroscientist and that's my particular area, I know that calcium is required for release of neurotransmitters in the brain and throughout the neuron. So while a lot of times we think the calcium is being used for something for the bones, it's actually just being stored and then going to be used by something else in our bodies. But for the projects I'm going to talk about, we're going to be interested in the phosphorus, which is typically stored with the calcium in the bones. I do include a link to a video, which if I had more time, I would actually show, but this shows the connection between the effects of weightlessness on astronaut bones. This was from a couple of years ago, so this is before the research that I'm about to show you. And I also include an instructional activity that focused just on the bones. This one in particular is a part of a series that we produced for high school students. It's called Math and Science at Work. It's a bit older, so some of the problems will be framed in terms of the space shuttle as opposed to the International Space Station, but being an AP biology teacher or biology teacher in general, the basic underlying biological concepts don't change. So this one is one of a series, and it focuses on calcium regulation and homeostasis in the bones. How does the bones uptake that calcium and store it and then release it at some point in the future, which typically tends to be several questions on the AP Advanced Placement biology exam here in the United States. There is one for the muscles, but I'm going to show you something different for the muscles. So that's your instructional resource for that. I then include also for muscles, sort of an overview, reminder what muscles do for us. Include a video that shows what happens to the muscles for astronauts when they're in space. Once again, for time considerations, I'm going to go ahead and skip over that. This is the instructional resource that the activity I'm focused on is for the muscles, but it actually turns out that this curriculum has both bone and muscle activities. Generally, it's for grades. This one is for middle school, upper elementary, and middle school. And there's one activity for the bone density and one activity for the muscle stress. Muscle stress is actually relatively straightforward and has the students get into pairs and get a clothespin, whether it's a wooden clothespin or a plastic clothespin, doesn't really matter. I sometimes use the little chip clips that I use to hold my potato chip bags. I actually forgot to go get one because Miss Ninja had me planted to the chair, so I forgot to get it before tonight. But anything where you squeeze and it opens, so clothespin is what's listed, but you can use anything like a clothespin. And within a particular period of time, one student is going to try to squeeze open the clothespin as many times as they can, while the other student is timing them. And the point of the idea here is at some point during that time frame, I think it's a minute, it might be 90 seconds, students are going to notice that their muscles are getting tired and they're going to slow down, and they may not necessarily be able to squeeze the clothespin as rapidly at the end of their minute or 90 seconds as they did at the beginning. And so the idea is to kind of to show that it's real easy to fatigue muscles, and if you don't use them, it's going to make it harder. So if the student were to, say, do the clothespin activity and, say, get like 20 squeezes in a minute, over the course of several weeks, if they were to practice, they probably could get more squeezes of the clothespin after 3 weeks or 4 weeks of practice. And so while it's not entirely a perfect representation of what happens to the muscles in space, it kind of helps the students to understand that if you don't use it, it's going to be a lot harder to do activities, and that's exactly what happens to the astronauts. If they don't use their muscles in space, it's really hard for them when they land and are brought out of the space capsule for them to stand, and that's part of the reason why we're interested in this as much as we are, because we want to make sure the astronauts are healthy and safe. So let's talk about real quick in the time that I have left, bone and muscle loss in aging and microgravity. If you're not already familiar, what happens to astronauts in space when they're exposed to microgravity is that there is rapid bone and muscle loss. If they don't use it, the body basically goes, oh, we have excess bone, we have excess muscle, we need to get rid of it, and this can happen very quickly. Even the shuttle missions, which were 16 days long, astronauts were exhibiting bone and muscle loss. So there's a lot of things that happen with bone physiology that even if I had the time tonight, I wouldn't actually have a chance to go into. But bone is living tissue, so it's constantly remodeling itself as a result of a variety of different things. Essentially, what happens to astronauts in space is accelerated aging. So what we experience here down here on Earth over the course of decades happens to astronauts, as I said, as quickly as over 16 days you can see effects, but certainly in the 5 or 6 months that our astronauts are spending on the International Space Station, if they weren't exercising, their bones and muscles would look like a 70 or 80-year-old human down here on Earth. So obviously this is a very serious issue. Taking a look at mouse models, it's been identified that as you lose bone, yes, you're releasing calcium into the blood, but you're also releasing the phosphorus in the form of phosphate ions into the bloodstream. And what ends up happening is those phosphate ions bond with some proteins to form calciprotein particles, CCPs. And thanks to these mouse models of human diseases that have been used down here on Earth, this class of molecules, particles, has been identified as possibly having some sort of a role in aging and possibly the accelerated aging that we see in humans and mice in space. So to tie this to current research, this is an ongoing set of investigations that are on board the International Space Station right now. You have that bone and muscle loss that is that accelerated aging. There are two complementary investigations. One is collecting data and tissue samples from humans. That's the phosphoaging project. And then you have a simultaneous project using the Astro mouse habitat 6, or MHU6. These are actually led by the Japanese space agency, JAXA, so they're being conducted on board the Kibo module. But just to give you a little bit more about these CCPs, they're actually nanoparticles that are the combination of these calcium phosphate precipitates, because they don't dissolve real easily in the blood. So they're kind of these particles that are floating in the bloodstream, and they form a complex with these serum proteins. And if you take a look at blood or urine, you can actually measure these CCPs, and the concentrations can change if you change the level of phosphate in the blood, and whether that's from an increased dietary intake or the kidneys not being able to get rid of the excess phosphate as well. If you have an increase in these CCPs in the bloodstream, what tends to happen in these mouse models of human aging is you get inflammation and arteriosclerosis in the blood vessels, and you have damage to the kidneys, fibrosis and tubular damage. This systemic tissue damage, anytime you damage tissues, you're going to trigger inflammation, and so it becomes this vicious cycle. And so the idea is, or the thought is that this triggering of inflammation by the presence of these CCPs in the blood is somehow contributing to and accelerating the aging process. So both of these projects are simply taking a look at the hypothesis that these CCP particles that are going to be released as a result of bone and muscle loss from the astronauts, because they're in microgravity, they're not using this, that this increased release from the bones is what's causing what we're seeing, this aging type syndrome that we see in the astronauts when they've been in space for any period of time. And so, as I mentioned, we're going to have some astronauts who are going to provide samples, blood and urine, at 8 different time points, both before, during, and post-flight. The pre-flight measures are going to serve as the controls upon which the in-flight and post-flight measurements are going to be measured. You've got your Astro mice, 6 of them got into space on this particular project. They were paired based on age and sex with ground-controlled mice on Earth. Basically, that just means the mice are roughly the same age in the pair and the same sex, and they're going to provide blood and tissue samples while they're in space, and all of that stuff then will be packaged and brought back down for analysis down here. But the idea is osteoporosis, osteopenia, the loss of the bone mass, is releasing this phosphorus. The elevated phosphorus in the bloodstream then creates a situation where you've got the increased level of CCPs, which are known to create inflammation in aging in the mouse models, and that is somehow triggering this vicious cycle that we see in the astronauts in space. If that hypothesis is in fact supported by the data that these astronauts and astromice provide, that will open up a whole new realm of pharmaceutical therapeutic interventions that would not only be useful to keeping the astronauts healthy while they're in space, either on the moon or on the way to Mars, but then there's that Earth-based application. Remember, that benefit to humanity. Those of us who are here on Earth who are going to go through the natural course of aging, this may potentially open up a way to help us retain, particularly females, because osteoporosis tends to strike us more frequently than males. This may open up a whole new way of treating osteoporosis, osteopenia, and keeping our bones, for those of us who are here on Earth, healthy, and kind of mitigate those effects of aging a little bit more. So, as I said, these are in progress, so I can't show you any data, but it is an incredibly exciting set of projects that, as I said, could open up a very intriguing line of biomedical therapies here on Earth to help treat those of us who age, and that's going to be all of us. So with that, I am at the end of my slides. I throw this in particularly for the university professors or those who teach upper high school. If you're interested in learning more directly about the nitty-gritty, the research, the in-depth research, remember I said I wasn't going to go into that because that wasn't necessarily the focus. If you are interested in learning more of the in-depth data, I direct you to our ISS Research Life Science Data Archives. All experiments in the life sciences that have been done, actually going all the way back to Mercury. All the results are archived at this particular. You just have to search by mission or experiment or something like that. So those of you who might want to learn more about the biomedical research that's been done in the past and ongoing, you might want to check out this particular resource. So with that, let me throw up my last slide. I first will provide you for anyone who may have questions afterwards. You can always contact me at my email. I don't always respond right away, but that's because there's a lot of you and only one of me. But I do respond usually within a couple of days of the time that you email me. So if you would like to, there's my email. Reminder that the post-event quality surveys will go out in the next 24 hours. Most of us, that will be January 28th. For those of you in England, Kenya, India, Thailand, other side of the world, basically, it'll be more like January 28th. January 29th, excuse me, be January 29th for you. You want to make sure that you fill that out in about a week. If you filled out the survey, and you must fill out the survey to get the certificate, that will come out February 3rd while I am doing my presentations at Seek. As I mentioned, recording link and webinar resources are available upon request, either in the chat box now or by email later. And although we will be off next week, if you're at Seek, you will obviously be able to get your EPDC knowledge fix if you come to any one of our presentations. But if you're not able to join us at Seek, either in person or virtual, we will be back with webinars the following week. Barbie will this is by no means a comprehensive list, but at least I give you a suggestion for each of the days next month, or excuse me, not next week, the following week. February 7th, Barbie will be talking about GLOBE and the cloud protocols. Tuesday, Dr. Samuel Garcia will actually be talking about launch integration and robots, I believe, with NASA from Earth to Space Part 2. And I did see a question about robots. I'll come back to that in a second. Dr. Deepika Sangam is going to be over the course of the next 2 months in preparation for that Artemis 1 launch that we hope will be March 12th or sometime in March and April. She will be starting Artemis Learning Pathways. She's actually going to be doing a role model for the Artemis Generation webinar on February 9th. And then I will be back to talk about the sun and its science and SDO, the Solar Dynamics Observatory, 2 weeks from tonight. So by all means, feel free to sign up for any of those if you'd like. And the early part of the program, I think it was Sylvie dropped in the link for our entire webinar schedule. So feel free to scroll back and get that and check out our other webinars. As I said, this is by no means comprehensive. And Miss Ninja, who has reoriented herself so that you can see her little spiky fur towards the top here. And I thank you very, very much for taking the time to join me and for your indulgence for a few extra minutes over so that I was able to answer your questions. And I will stick around and answer any more questions that you might have. And we may have or have asked. I'll stick around for a couple of minutes. But in the meantime, please stay safe. Hopefully I'll see some of you perhaps next week. I am presenting virtually. I will not actually be at Seek, but I will be virtual. So hopefully I'll see maybe some of you next week as part of the conference. Otherwise, I will see you all in 2 weeks. And of course, because I'm doing it virtually, Miss Ninja will be with me at Seek. So she doesn't realize it yet, but she gets to go to Seek with me, virtually at least. So have a great day wherever you are in the world. Let me scroll back and see if I can find questions that I might have missed. Okay, so I got the how young are the mice. Okay, for the fourth grader. Okay, cool. I got the reproduction one. Okay. Okay, so I promised Suari that I would come back, and I'm not sure if I pronounced that right or whatever. Suari asked in the program how animals survive safely to their trip to ISS without spacesuits and how they behave to microgravity effects once they're on board the International Space Station. This is a great question because when I was a kid, I used to draw mice in Astro suits because I figured they had to be in Astro suits in order to get into space. It turns out that their transporters and the cargo ships more importantly, the cargo ships or the crew quarters that they travel are pressurized and have the atmosphere and the food and everything like that so that they're able to function without actually having to wear spacesuits. So when I mentioned in the ConOps, the concept of operations at the beginning, that is all part and parcel. They're set up in their transporter. They'll have all the food and water and amenities that they might need, because obviously they're not going to stop being mice just because they're inside a transporter on the way for the International Space Station. So their cages, their transporters are designed in such a way to be able to handle them for a period of time in that cage, where they'll have the oxygen, they have the water, they have the food, and all of the waste products are taken care of. They're usually inside. If they're traveling with a crew dragon, you don't necessarily have to worry about it, because obviously we're going to have the capsule pressurized and aired and everything for the astronauts. So the mice are in there with the astronauts; they're taken care of. I believe in the cargo dragon, that also will be have some sort of environmental control. Now, animals don't necessarily ride on every single resupply mission. They ride on most of them, depending on the research schedule. But usually it's pressurized. There's some sort of environmental control in the cargo ship, even if there's no astronauts on board, just because we want to make sure that they're taken care of in the absence of spacesuits. So hopefully that answers your question. Okay, let's see. So Fazio asked during the program, what about sending robots? And Fazio, that's an excellent question. And we're actually doing quite a bit of that. So a lot of the pre-work in going to the moon or going to Mars has been done by robots. So your curiosity, your perseverance, your insight, spirit and opportunity, those sorts of things are laying the groundwork. And certainly robots are, we can send them into conditions that are very much inhospitable to humans or mice or any sort of biological creature. And so you have that option. At a certain point, though, humans are going to want to go. Maybe not all of us, but some of us, that spirit of adventure, that spirit of exploration are going to want to go. And so that's the idea, is we want to make sure that everything is safe. So robots, yes, factor very heavily into sending them in and doing some of those things before humans ever get there. So in terms of building the moon base, you're probably going to have a lot of robotic activity on the surface of the moon long before humans are ever there. That the robots will be on the surface building and preparing the habitat long before the astronauts are actually there. Same thing will happen with Mars. So in terms of setup or going out into the environment for the first time to test it and see what's going on, yes, robots are. But at a certain point, humans are going to want to go. And so at some point, you've got to figure out, okay, how do we get humans there in a safe manner? And so one of the other webinars that you might be interested in learning more about and what NASA is doing to prepare for these deep space missions is a recently released curriculum, Hazards of Spaceflight to Humans. It's a 5-part series, and I think Deepika and Stephen and possibly Samuel do webinars based on that curriculum. As I said, it was just released in advance of the Artemis 1 launch. What are the hazards that we humans are facing when we talk about living on the moon or going on to Mars? So hopefully that kind of helps a bit. Kelly twin study, yes, really cool. Learn about the impact on humans. Yep. We actually have had, in some instances, astronauts who are staying up there for a period of time. Mark Vanderhey is up there right now. His mission is slated to actually go a little bit longer than Scott Kelly's, but Mark doesn't have a twin, so yeah. The Kelly twins still kind of get the cool factor because of the fact that they were twins and astronauts, and you were able to have that ground control with Mark and then Scott be up in space. Okay, let's see. How long have the mice in MH6 been in, will be in space? Patrick, they could be up there as long as 6 months, but I don't think they're going to be up for the entire 6 months. I think they went up in November. I think. I'd have to do some research. I didn't actually look into the details of when MH6 actually went up. And it could be sort of an ongoing. So you got six astromice go up at a time, they come back down. Another six astromice go at a subsequent mission, come back down. So I don't think any particular set of six mice are up there for longer than 6 months. It really depends on the comings and goings of the cargo ships. So I know that I believe a SpaceX cargo Dragon just came back from the International Space Station, and it may very well have been carrying some or all of the MH6 mice on board. So this is January. If they went up in November, that's about 3 months. So it could be anywhere from 3 months to 6 months, depending on the coming and goings of the crew or cargo dragons. Okay, thank you. Thank you. You're very welcome. Glad when we have the young students join us, and glad they enjoy it. I always love doing the life science ones because I love animals, first of all, but animals engage students like nothing else. It's amazing. I have this little thing that I typically share when we're on center. Obviously, we're not on center right now because of COVID, but we have a variety of animals, and I've done this in a couple of my webinars and a couple of my face-to-face presentations. I will tell students that these cats or deer or squirrels that are on center at Langley, they're not just any cats or deer or squirrels. They're space cats or space deer or space squirrels. So pretty much any organism that we find on center gets dubbed a space something. Now, we've had a group of Canadian geese show up. They're now space geese. And the hysterical thing is the teachers always come up to me and they're like, 10 years from now, these students will remember nothing else other than the space geese or the space squirrels. So, but yeah, students love the astronauts. Very welcome. Very welcome. Have a great evening. Not sure if you're all here still, but Eliana, thank you. Marcus, thank you. Llewellyn, thank you. Umura, I hope I'm pronouncing your name right. You're very welcome. Faucio, you're welcome. Lynette, you're welcome. Sylvia, oh yes, Miss Ninja does get free attendance. Of course, Miss Ninja is completely oblivious to the fact that she gets to go to the conference, at least now. Obviously, whenever I start talking to the computer, she knows that that is time to go greet the humans. So when I start talking next Tuesday, or not next Tuesday, next Thursday for my first presentation, she hears me talking. I'm willing to bet she'll be up here real quick if she isn't already up there by that point. Sylvia, yes, do they restrain the mice during takeoff and landing? To the extent that they are in the transporter, that's, I believe, how they restrain the mice. There's no like with the astronauts, you have like the fitted cushions or the seats, cushion seats or the couches or however you want to talk about it in the space capsule. Yes, they have those specially designed chairs for the humans, but for the mice, I don't think there's really any restraint. They might have designed, and I didn't have a real good picture of the transporter. There's up to 20, depending on the mission, there may be up to 20 mice in there. And just the fact that they're in there might prevent them from moving too much. But when you have that many mice, I don't think there's really any way that you can like put seat belts on them or some sort of restraining belt to keep them from moving. I think they're just kind of in the cage, loose, small space, so they can't move around too much. But I don't think there's too much ability to restrain them in the way that like we wear seat belts or built in the astronauts on the capsules. But it's an excellent question. I'll have to double check and make sure. So Patrick asking, just curious if there would be a mission with an ongoing mouse mission to space, lifetime in microgravity, for example. And that may very well happen. I earlier in the program, I mentioned that the mice usually are up there 3, maybe 6 months because of the particular scientific experiment. But there's nothing to say that perhaps as we get closer to becoming a truly spacefaring species, that we might not have some experiment where we have mice, for example, and we basically watch them through their entire life cycle to see what exactly happens to them, so we get some idea of what might happen to humans that spend their lifetime in microgravity. That obviously is, I believe, very far in the future, so think more like space science fiction at this point. But there's nothing saying that at some point, depending on research question and goals and objectives of the research in what we're doing for a human space program, that might not come to pass. I don't think there's been anything yet that approaches that. I think usually the mice are up there for a shorter period of time. I don't think any mice have been up there for their full lifespan, but certainly something to think about for the future. After hours mysteries. Yes, I will probably get a lecture at some point. You all who are regulars know that it's closing in on 8:30 on the East Coast, which means I'm about 20 minutes late with dinner. So yeah, I will get a lecture about how I'm delinquent in my feeding duties at some point here. She is still kind of sleepy. We have been battling a cold the last 2 weeks. I think we're starting to round the corner. Of course, cats don't have the best immune system, so she's been awfully dopey and sleepy the last couple of weeks. And she's been sleeping most of the time. She's going to get up and turn here, but she's been sleeping most of the time the last 2 weeks. But once again, she's 17 years old. She's kind of allowed. But yeah, don't worry. I will get a lecture. I will get the stink eye. I will get something at some point. So you're very welcome. Thank you for those of you who've stuck around. I will stick around for a couple more minutes. If you have any questions or comments, or just enjoy Miss Ninja. But yeah. Yeah, I joke. I'm just here to handle the feline education specialist. Everybody's here, for the cat. See ya. Have a good evening. I will. I will. I'm actually taking a couple of the webinars that I've done over the last year and kind of doing a more in-depth version of them for the conference. So feed the cat. Oh yes. Yes, although with the cold, she's not been eating as much, so it'll be really interesting once she gets past the cold. She'll probably be eating me out of house and home. Have a great evening. Kibble last while. Yes, yes, yes. That's what saves me. I'll put out something, Fancy Feast, or her favorite skipjack tuna. I'll put that out, or blue. That's the other one that they eat. I'll put that out, but if she totally inhales it, yeah, kibble saved me, because if she's still hungry, there's still a bowl of kibble, so make sure that's always filled. Kind of. Kind of. I was able to get not so Fancy Feast makes two kinds of skipjack tuna. The purely Fancy Feast is the one she really loves. I was able to get the other one, which is okay, but not as good. So, as I said, kind of. We seem to be having some supply chain issues, not only with human food, but cat food, because everywhere I've gone to include Chewy.com, it's completely out of skipjack. So I was able to come up with something that was still Fancy Feast and still skipjack, but wasn't quite as good as the usual stuff. Once again, I got a lecture. But yeah, so Santa kind of delivered. I need to stop the recording so it can buffer. Absolutely, it's all in that master class. So, yeah, they've been advertising Neil deGrasse Tyson, how to think scientifically, Joan Benoit Samuelson, how to run, Miss Ninja, how to deal with space humans, master class. This is her master class. Meow. Let me go ahead and stop the recording so we can start buffering.